Okay. Hi everyone, welcome back to Neuroscience for Non-Majors. This is week 5, unit 4, part 1. I decided that I'm going to split the sleep and memory unit up into 2 parts. Today's part will focus on kind of how memories are encoded and formed, specifically declarative episodic memories in the hippocampus. And next week, we'll talk more about long-term memory and sleep, which are very much connected to and relevant to each other. Yeah, so I'll share my screen. There we go. Yeah, so this week's lecture topics will include the hippocampus and entorhinal cortex, place cells and grid cells, types of memory, hippocampal indexing, and engrams and optogenetics. So the hippocampus, meaning seahorse in Latin, is a structure that is embedded in the temporal lobe. We have 2 of them, one on either side of our brain. And the hippocampus is divided into different parts and connected to a structure called a subiculum, which is connected to a part of cortex called entorhinal cortex. The parts of the hippocampus are the dentate gyrus, which means looks like a tooth in Latin, and it contains a lot of neurons, and the cornu-ammonis, which it means looks like a ram's horn. It's kind of like curved like that. And the dentate gyrus again contains a lot of neurons and they're thought to be only like it's one of the only places in the brain where new neurons are formed. So like adult neurogenesis as an adult, lots of neurons are formed when you're like a baby or a fetus, but when you're an adult, the dentate gyrus is one of the few places where baby neurons are still being born. And the cornu-ammonis is divided up into different subfields, CA1, CA2, CA3, and CA4. Okay, so there are specific names for the different axon projections between various parts of the hippocampus and entorhinal cortex. The projections from entorhinal cortex to dentate gyrus are called the perforant path, and the projections from dentate gyrus to the cornu-ammonis subfield 3, CA3, are called mossy fibers. And then the projections from CA3 to cornu-ammonis subfield 1, or CA1, are called Schaffer collaterals. In terms of navigation, the entorhinal cortex contains these things called grid cells, which fire at regular intervals throughout the process of spatial navigation and create a sort of grid map of the space that you're navigating. This is occurring alongside a process in the cornu-ammonis, which allows you to pinpoint your location within that grid map. Cornu-ammonis subfields 3 and 1, CA3 and CA1, contain pyramidal place cells. And these are spatially localized cells, meaning a portion of them will drastically increase their firing rate if you occupy a specific location, and a different portion of them will drastically increase their firing rate if you occupy a different location. And the specific location that kind of triggers this high frequency firing in a portion of the CA1 or CA3 cells is called the cell's place field, or it's called the place field. So memory can be divided into 2 main categories of declarative and procedural. Declarative memory, which we'll focus on for today, is memory that deals with conscious recall of facts and past experiences. It's the explicit memory. Procedural memory, on the other hand, is implicit memory and does not require conscious recall. This type of memory dictates our subconscious processes, and we will learn about procedural memory more in a future lecture. So Brenda Milner was the scientist who kind of showed a lot of information about declarative and episodic memory, specifically through working with a patient named Henry Molaison, who had his hippocampus removed, and she kind of showed the distinctions between a lot of memory that was declarative and a lot of memory that was procedural, and which kinds of memories and needed the hippocampus and which ones didn't, and things like that. So it's very interesting. But the hippocampus is specifically crucial for forming and storing short-term memories, and that ability to kind of create new memories was disrupted in that patient, Henry Molaison, who had his hippocampus removed again because he suffered from epilepsy and he had to get it removed. So the mechanism by which your memories are encoded and your experiences turn into your memories, into short-term memories, it's called hippocampal indexing. And so basically during an experience, all sorts of areas around the brain are active, right? So all of these areas are sending inputs to the entorhinal cortex during an experience. And while it's not immediately relevant, the entorhinal cortex also has projections back to all of these various brain areas as well. So they have like, so various brain areas project to the entorhinal cortex, and the entorhinal cortex also projects back to those various brain areas. But anyway, so various brain areas are sending inputs to the entorhinal cortex during an experience. And if this experience is going to be stored as a memory, then the entorhinal cortex will activate a unique group of neurons in the dentate gyrus through the perforant path. And as the entorhinal cortex is getting all of those inputs from the areas of the brain that are currently active, the dentate gyrus has enough cells that it will be able to activate a unique combination of dentate gyrus cells for every experience. This unique group of cells corresponding to the experience is called an engram. And the dentate gyrus engram activates another engram in the cornu ammonis subfield 3 through the mossy fibers. And so CA3 is really special because it has a lot of reciprocal projections within itself. So all of the neurons in CA3 are like very much connected to each other, both as presynaptic and as postsynaptic neurons. So long-term potentiation is also incredibly important in the hippocampus because all of these connections and interconnections and reciprocal connections. So and the strengthening of synapses due to simultaneous activity, which again we learned was long-term potentiation, can last for a year in the hippocampus. Okay, so now we have an engram in CA3, which was activated from an engram in dentate gyrus, which was activated from the entorhinal cortex, which was activated from the experience. Now, later on, if a portion of that original experience is present, a reminder, so like instead of all of these inputs, it's just like a small portion of the inputs, then those areas of the brain are going to reactivate the tiny portion of the original experience, and it's going to activate a tiny portion of the entorhinal cortex cells that were active during that experience, which will activate just a tiny portion of the dentate gyrus engram portion. Like again, over here, if this was a full engram, then this is like a tiny portion of the engram that's activated in the presence of a reminder. Then that tiny portion of the engram activates a tiny portion of the CA3 engram. But then again, remember, because of all of these interconnections in CA3, if just a tiny portion of it is reactivating, then the entire CA3 engram is going to reactivate. So this tiny bit of the experience, a reminder, is going to end up reactivating the entire experience's engram in CA3. This activates an engram in CA1, the full engram, and through the subiculum, it's going to reactivate now all of the areas of entorhinal cortex that were active during the original experience. And because of those reciprocal connections back to the various brain areas that entorhinal cortex has, again, the entorhinal cortex will now, because of this full engram reactivation, go back and reactivate all of those various brain areas that had originally given it inputs during the original experience. And this is a memory, the reactivation of all of those same brain areas that were active during your original experience once you see a reminder. So I think this process is really, really cool. Now, in the past decade, a lot of research into engrams and this type of declarative memory regarding your autobiographical timeline called episodic memory has been conducted through a technique called optogenetics, which we're actually able to do because of green algae. So green algae unicellular organisms move in response to light in a process called phototaxis because of channel proteins in their membranes that open and close in response to light. These proteins are called light-activated ion channels, and the specific one used in optogenetics most commonly is called channel rhodopsin, which responds to blue light. When channel rhodopsin absorbs a photon, then it will change shape and allow ions to pass through, including sodium and calcium and magnesium ions, which again really want to get into the cell, the neuron. So now how can this green algae ion channel help us study memory? Let's backtrack for a second and think about how are proteins even created? Well, the nucleic acid called DNA contains genes or specific sequences of sets of molecules which attract other molecules that form complementary sequences in another type of nucleic acid called RNA. And when this complementary RNA sequence is created, it goes to something called a ribosome, where more RNA molecules arrive, carrying amino acids with them. And as these amino acid carrying RNAs stick to the genetic information carrying RNA, then the amino acids start forming bonds with one another. And as they do, they release from the RNA that was carrying them, and these strings of amino acids then fold up to form a protein. Now, different proteins are formed at different times and have all sorts of different purposes, as you know from studying the different enzymes and channels and receptors we've looked at so far. There are certain proteins coded from genes called immediate early genes in neurons, and these proteins are directly tied to neural firing. So they will be coded a lot more, and they'll like appear and be active as soon as a neuron fires. So like they're directly correlated with neural firing, and they'll only be active only when the neuron fires. Two of these IEG proteins are ARC and CFOS. And now experimenters in the early 2010s did something really, really ingenious with a virus and a specially constructed gene sequence that involves channel rhodopsin and immediate early genes and something called Cre recombinase, which I'll talk about. But I'll briefly talk about how a virus works too. So the way a virus works is that it's kind of a blob of proteins and membrane that contain genetic material, nucleic acids, either DNA or RNA. And the reason it can infect cells is because the proteins that it expresses on its surface membrane are the same proteins that our cell membranes express. So the cell membrane is likely to let the virus bubble fuse with it, which consequently infects the cell with the genetic material that was inside the virus bubble. And the genetic material then codes for proteins using that process we just went over. And in nature, more virus proteins are coded, and they then like bud out of the cell and infect other cells. But scientists have begun to use viruses as a tool to inject cells with genetically engineered nucleic acids, and optogenetics is one of the ways you can use a virus as a tool for. So what scientists did was create a gene sequence that had the CFOS gene, again the immediate early gene, then a gene for an enzyme called Cre recombinase, again, I'll talk about in a second, and then a gene for channel rhodopsin. And also they would have another gene that has a fluorescent protein attached after channel rhodopsin as well, so that they could identify, like, to see if the gene sequence was actually being like expressed. So there would also be like a fluorescent protein gene in addition to that. So Cre-recombinase. If the enzyme Cre-recombinase is in the presence of the substrate that activates it, then it has the ability to actually cut out portions of DNA and reattach the strands together without the cut out portions. So you can use it to actually get a specific gene to be expressed at a specific time by putting some like junk sequence of DNA in the sequence that can only be cut out when Cre-recombinase is activated by its substrate. So, okay, put that CFOS, Cre, channel rhodopsin, green fluorescent protein gene sequence into a virus bubble, a viral vector, and then inject the virus into the hippocampus of like a mouse. Now the cells in that mouse hippocampus will contain that gene sequence, which has the light activated ion channel rhodopsin in it, that will only be activated if both the neuron is firing and Cre-recombinase is active. So yeah, again, if this is a mouse, for example, you can actually inject the mouse with the substrate for Cre-recombinase. They usually inject it, I think, with tamoxifen. And this mouse, again, has the virus in its hippocampus, so it's expressing this gene sequence. And so now, then you can put the mouse into a context where it gets like some air puffs in its face, which mice don't really like. And you can do this until the tamoxifen wears off. And so what will this do? Well, all of the cells that had been active and expressing the immediate early gene CFOS for the duration of the air puff experience were also expressing the proteins coded from that genetically engineered sequence. And since this was all in the presence of the tamoxifen substrate, Cre-recombinase was able to cut out junk DNA, blocking the channel rhodopsin gene, and then the channel rhodopsin was expressed in all of the cells that were active during this experience. In the hippocampus, these cells would be the engram for that experience. So now we have tagged the specific group of cells that represent and can trigger an episodic memory with this. We've tagged this group of cells with the light activated ion channel. So the next thing to do then is to, through a fiber optic cable that goes into the hippocampus of the mouse, see what happens by shining light on this engram in a different, like safe, non-air puff context. And actually we see that when we shine light on the engram, then the light-gated ion channel proteins will open and positive ions will flood into the cell and cause them to fire again, triggering the experience or the memory again. So the mouse actually shows fear and discomfort in this new, like, cozy environment, even though it has never been in the environment before, because we're making it, through the light shining into its brain, re-experience the air puff memory. So I think it's really cool how we can literally control the firing of neurons with light through this technique of optogenetics. It's pretty interesting. All right, so that's it for today. And for homework 4, I want you to write an outline for like a short, like as short as it takes to demonstrate understanding, horror movie involving and demonstrating your understanding of at least 3 of the following topics: hippocampal indexing, engrams, place cells and grid cells, channel rhodopsin, and optogenetics. And yeah, these were the references. I think this is a really interesting paper that it kind of goes through hippocampal indexing. And I think it's really interesting and you should read it if you're interested. But yeah, I'll see you soon.